Welcome to another video on Biology by Perfect's course. This is Preetinder Kaur, and the important topic that we are going to cover in this video is that of meiosis. So before we get started, don't forget to visit us at perfect-scores.com, like and share us at Facebook, and give us your valuable suggestions at Gmail. So what exactly is meiosis? Meiosis is simply known as a reduction division. Why? Because it reduces the number of chromosomes in the cell by half. So that is why it is known as a reduction division. So basically it helps to convert a diploid nucleus, which is 2N, into a haploid nucleus. So this is diploid. And talking about the numbers, one diploid cell gives rise to 4 haploid daughter cells. And basically this process takes place in the sex organs, and this is the process by which the gametes are made in the reproductive organs. So the gametes are made in the reproductive organs. Now most sexually reproducing animals are diploid. For example, human beings. We have total 46 chromosomes, out of which 23 are maternal and 23 are paternal, or ones that we have got from our father. Similarly, maternal is the one that we have got from our mother. Now that means we have 2 copies of every chromosome. Now in order to reproduce, such organisms need to make gametes that are haploid. That means have only 1 copy. So diploid cell would be having 46 chromosomes, whereas haploid cells that you will get will be each having 23 chromosomes. So fertilization of the 2 haploid gametes. So let's suppose this is one gamete and it fertilizes with another gamete that also has 23 chromosomes. This will later result into formation of a zygote, which is going to have again the normal number of chromosomes. So this zygote is going to be diploid, and that will grow into a new organism. So coming back to the structure and the process of meiosis. Meiosis, it consists of 2 major stages. One is known as meiosis 1, and this is the reduction division. So you need to know all these major points. Where does reduction division take place? And we'll be more specific ahead. So right now you need to know that meiosis 1 is where reduction division takes place. So from one diploid, you form 2 haploid cells. Alright, so that is going to be meiosis 1. The next step is meiosis 2, which just separates the sister chromatids. It is just like mitosis. But this division is necessary because meiosis is preceded by the interphase wherein DNA is replicated. So that means since DNA has become doubled, the sister chromatids need to separate, and this is what happens in meiosis 2. So from each you get 4 more. So the first one is meiosis 1, where from diploid you get 2 haploid daughter cells, and meiosis 2 is when from each haploid daughter cell you get 2 further haploid daughter cells. So let's go ahead and get started with this process, but before that you need to know an important concept in the area of chromosomes, which is homologous chromosomes. So as the name suggests, they are quite similar to each other. So homologous chromosomes are basically chromosomes that share the same structural feature. That means their size, their breadth, everything. They have the same size. They have the same banding pattern. And they have the same centromere position. So I hope you remember what is a centromere. So if this is a chromosome, this point over here in the middle is known as a centromere. Along with same structural features, homologous chromosomes also have the same genes at the same locus positions, at the same loci or at the same positions. Now the genes are same, but the alleles may or may not be different. So let me give you an example of a tall and dwarf plant once again. So let's suppose there is an organism that has a total of 8 number of chromosomes. So that is the diploid case. That means it has got 4 from the mother and 4 paternal from the father. So as you can see, these chromosomes are always arranged in pairs. So these chromosomes, for example, they look like the same. That means they have the same structural feature. Even this pair of chromosomes has the same structural features, same for this, same for this. So this is a pair of homologous chromosomes. Also, they have genes at the same position. Let's suppose the gene for the height of the plant is located on pair number 3. Alright, that's a hypothetical case. So let's suppose the gene is located at this area of chromosome in the third pair. Then in the corresponding homologous chromosomes, it will be located at the same position. The alleles may be different, but the gene will be present at the same position. Similarly, you can have a gene for something else over here and here. There could be another gene at these corresponding locations, another gene over here, so on and so forth. So these are going to be the homologous chromosomes. Now let's go ahead and begin with the process of meiosis, including the pairing of homologous chromosomes and crossing over, which is a very important process. Now, the process of meiosis involves 2 divisions, both of which follow the same basic stages as mitosis. That means you have a prophase, then you have a metaphase, then you have an anaphase, and then you have a telophase. Meiosis is preceded by an interphase, which includes the replication of DNA. So let me put it here. This is where replication of DNA occurs, specifically in the S phase. And this helps to create chromosomes that have genetically identical sister chromatids. Now, what are sister chromatids? Let me go back to the previous slide. Now, a chromosome over here is actually divided into 2 parts. The part on the left, let me color it with this orange color. This is known as one chromatid. And the part over here with the dots is the second chromatid. So these are basically 2 parts of the same chromosome. And because they're parts of the same chromosome, we call them sister chromatids. So sister chromatids are nothing but chromatids that are attached at the centromere to each other. And during one of the divisions in meiosis, we will see how they get detached from each other. So let's begin by the first process. So these are the 5 steps that we are going to study in meiosis 1, which is interphase 1, prophase 1, metaphase 1, anaphase 1, and telophase 1. So the first phase is interphase 1, and as you can see, it has a proper nuclear membrane. So I'm going to write down the main things at the bottom. Nuclear membrane is there, and all the genetic material is in the form of a dense network of chromatin fibers. The next process is going to be prophase. Now you will observe in prophase, the nuclear membrane starts to disintegrate. Additionally, you will be able to observe that the centrioles, they place themselves on the opposite poles of the cell, and the cell begins to divide by meiosis 1. Now I'm assuming that there are only 4 chromosomes. That means 2 pairs. So I'm going to write down the number over here. So right now the cell is still diploid. There are total 4 chromosomes and the centrioles are there. The nuclear membrane has disintegrated. Now one thing you will observe is this area over here. where sometimes the chromosomes, their chromatids of the homologous chromosomes, they come next to each other. So the chromatids, they sometimes appear next to each other in the homologous chromosomes. So there is a particular phenomena that takes place which is known as crossing over. A very important phenomena because that is responsible for the variations that we show. Now let me show you, for example, how does it actually happen. So let's suppose the blue chromosome is like this. Alright, and now let me just change the color to yellow. And the yellow chromosome was like this. Now what will happen at this point is these segments that are overlapping, they will cross over to one another. So what you're going to get is this. So there is no crossing over happening at the top, but there is some crossing over happening in the bottom of the chromosomes. So that means basically a segment of the yellow chromosome will go and get attached to the blue chromosome, and similarly a segment of the blue chromosome will get attached to the yellow chromosome. So this is known as crossing over, a very important step that happens in prophase 1, and the point at which the crossing over happens is known as a chiasmata. So that is the main thing that happens in prophase 1. Alright, so what happens in the next phase, which is metaphase 1, is these chromosomes, now the nuclear membrane has totally vanished. The centrioles have given out their spindle fibers, and what these chromosomes have done is they have arranged themselves on the equator. In total, there are still 4 chromosomes. Now let's see what happens in the next phase. In the next phase, in anaphase, what happens is now these blue and yellow chromosomes, they are going to get separated. That means 2 chromosomes will come on this end and 2 chromosomes will come on this end. And remember, these 2 bigger chromosomes, they've already undergone the crossing over. Alright, so this is what is happening in anaphase 1, and this is exactly where the reduction division will take place. The homologous chromosomes, they have gone in opposite directions, and as you can see, anaphase is nothing special, but in metaphase, the chromosomes are arranged on the equator, and in anaphase, they start moving towards the opposite poles. So let's see what happens in telophase. Now, as the name suggests, and as the shape is showing, the cell will actually start to divide into 2. That means the cytoplasmic division will begin, and on each end, the nuclear membrane will also start to appear. And each part is going to have 2 chromosomes. So that means these 2 daughter cells are going to be haploid now. Alright, so as this figure shows, the chromosomes have again become into the chromatin material, which is denser, and they're not thick anymore, and they have come and arranged themselves. The spindle fibers have vanished, and the nuclear membrane starts appearing. Now what happens is cytokinesis is going to take place and divides the cell into 2 daughter cells. Now similar process, just same as mitosis, is going to take place in the next phase. So let me go ahead and do that. I'll just draw out what are the different phases of meiosis 2. Alright, so I've just gone ahead and drawn these phases in short. So we begin with the results after the cytokinesis has taken place. Now you have 2 haploid cells. So this contains n number of chromosomes, where n was 2. And these are both haploid. Now prophase 2 happens in both of them. So as you remember, in meiosis 1, the crossing over had taken place. That is why this blue chromosome is showing a yellow part, and the yellow chromosome is showing a blue part. So in prophase 1, the nuclear membrane starts to disappear, and the centrioles, they come on the opposite poles of the cell, and the cell begins to divide. In metaphase 1, the nuclear membrane has totally gone away, and the chromosomes, they come and arrange themselves at the equator at the spindle fibers that are released by the centrioles. And then in anaphase 2, this is where the splitting apart of the chromatids takes place. So the sister chromatids, they split up because of the DNA replication that had taken place, and they start moving towards the opposite ends, and in telophase, that is the beginning of cytokinesis, and you can already see 4 haploid cells that are being produced. So right now it's N everywhere, and in anaphase 2, N number of chromosomes, it's at the different poles. So this is the end of meiosis 2. So I know it's a little complicated process, but the best thing that you can do is try drawing diagrams on your own for meiosis 1 and meiosis 2. So let's just quickly revise what we have done so far in meiosis 1 and in meiosis 2. So meiosis, it's preceded by interphase, which includes replication of the DNA to create chromosomes. And in meiosis 1, let me go back to the previous slide. Yes, this is meiosis 1. So the homologous chromosomes must pair up to be sorted into separate haploid daughter cells. In prophase 1, these homologous chromosomes underwent synapsis or the crossing over, and then they are held together at the points called chiasma, and the crossing over, it takes place, resulting in new gene combinations. So this result into new gene combinations, it is also known as recombination. So this is where recombination takes place. By chance you get a question on that. The remainder of meiosis 1, it involves separating the homologous chromosomes into separate daughter cells. So in metaphase 1, the homologous pairs, they line up along the equator. In anaphase 1, the homologous chromosomes split apart and move to the opposite poles. In telophase 1, the cell splits into 2 haploid daughter cells, and cytokinesis happens concurrently. Coming to meiosis 2, the sister chromatids are divided into the separate cells. In prophase 2, the spindle fibers reform and connect to the chromosomes. In metaphase 2, the chromosomes line up. In anaphase 2, this is where the separation takes place. The sister chromatids split apart and move to the opposite poles. And in telophase 2, the cell splits into 2 as the cytokinesis happens concurrently. Now, because sister chromatids may no longer be genetically identical because of the recombination, the process of meiosis, it helps to give rise to 4 genetically distinct haploid daughter cells. So each of these 4 is not similar in terms of the genetic construction. They're all different. So the next important thing that is quite related to meiosis is a disease, or rather a situation known as nondisjunction. Now this occurs when the chromatids, they fail to separate. It refers to the chromosomes failing to separate. So it's a small but very important part of meiosis. They fail to separate correctly. That can result in gametes with 1 extra or with 1 missing chromosome. So this difference, this deviation from the normal, is known as aneuploidy, aneuploidy. And the failure of chromosomes to separate, it can either occur when the homologous chromosomes, they fail to separate, when homologous separation does not take place during anaphase 1, or it can take place when the sister chromatids fail to separate during anaphase 2. So these are the 2 conditions under which non-disjunction can take place. One classic case of aneuploidy is the Down's syndrome. So what happens in Down syndrome is the cells, they have an extra 21st chromosome. So that means in normal individuals, there are only 2 chromosomes of the 21st pair, but in case of Down syndrome, they have 3 chromosome number 21. So this presence of 3 chromosomes is specifically known as trisomy. And this occurs as a result of non-disjunction. And that means one parental gamete was normal. So one was having only one 21st chromosome, the other was having two 21st chromosomes, and that gave rise to a zygote that had three 21st chromosomes. It usually occurs in women who have children above a certain age. So basically, when the two gametes fused during fertilization, the resultant zygote had three copies, and this leads to Down syndrome. So that is all that we'll be covering under this video for meiosis. It's a long video, and as I told before, the best way to understand meiosis completely is to keep drawing diagrams a few times, and then you will see that it's going to become very easy for you. So in the next video onwards, we are going to discuss more about a very important process known as karyotyping. So thank you so much for watching this video.